Hi, this is Garrett from the Gen3 team. I'll just give a quick introduction to our folks and then hand it off so that we can get started. On the line, actually, if you don't mind going to the next slide, Victoria. On the line, I have Victoria from our user services team who's going to walk us through kind of the user's perspective of using Gen3 for those who have a little less an opportunity to experience it in the past. Then we have Fan and Dimitri, two of our bioinformaticians who are going to walk through an example of an analysis done on Gen3 just in a Jupyter notebook so that we can see a little bit of a real life context. We'll hand it off then to Alex Vantel from our technical team who will discuss some of the technical perspective for Gen3 so that those on the line who are more development focused will have a better understanding as we talk through some of these interoperability questions, areas of interest for them and areas of focus that they should take. And I will, time permitting, circle up and essentially say everything that Brian just said in a very Gen3 specific context at the end if there's time. So without further ado, I'll hand it off to Victoria. And Victoria, in case you're speaking, we can't hear you.
Victoria's muted.
Yeah, Victoria, it does look like you're muted in Zoom. The best part of being the first people up is technical issues. Victoria, it looks like you are unmuted now. Can you?
Hello, can you hear me now?
Yes.
Yes.
Okay, great. I'm sorry, I wasn't able to unmute myself. So thank you, Garrett, for introduction. And I will, can you see my screen now?
Yes.
Okay, great. So I want to show you a typical user experience for data discovery in Gen3. And we will go through several steps, log into Gen3. I will show you a welcome page with information and quick links, exploration page for searching and exporting data, and queries and programmatic data exploration for experienced user. And I want to show you it like click around. So we just get started with a login page. So users can see one or several authentication options. Here on NCI CLDC, we see button login from NIH, but on other data commons, there could be just Google login or Microsoft login or any other identity provider that data commons support for this particular commons. So the workflow is very simple. You just click on Google login, you go to your identity provider, and browser automatically redirects you to Gen3 front page. And on the front page, user can see several blocks. Here we see a small introduction about data commons, and also we see overview chart. And this overview chart can differ from commons to commons. Here we see number of subjects, number of studies, number of samples, and number of imaging files, because these commons contain open brain data. But for example, on other commons, we can see instead of imaging files, we can see number of aliquots or other information. Here you see 3,000 aliquots. So this chart can be different from commons to commons. And also you can see different links, quick link to explore data model in data dictionary, explore data, query data, and run analysis. For our submitters, we'll also have a submit data button. So let's click on explore data model. And it will show you this nice page where you can zoom out, zoom in, and look at data model. It is graphical representation of Postgres database. And graph data model allows users to understand data structure quickly. For example, if we click on sample, we immediately see the path. Sample belongs to subject, subject is part of the study, and study is part of the project, and project belongs to program. And also, if we can click open properties, we see all properties that researcher can expect from querying this table. Another page for data discovery is exploration page. And this page also will look different on different commons. For example, here we have 2 projects, we have different subjects, and if you see slider here, we can specify age, and visualization will reflect immediately our selections. On other commons, you can see different facets. For example, here we have data with controlled access. So I am able to search data on the way I have access or without access. Without access, it will be only generic information for me. And with access, I will have more specific information and I can select everything. And there are several options to save my results. Depending on Gen3 setup, user have option to download all metadata or download manifest containing information about files or export to Gen3 workspace. Also, we have option to export to the Terra and export to PFB, which is portable format for biomedical data and is useful to export big amounts of data and import it to different commons. Another page for data discovery is our query page. And here I have a small example. For example, I am interested in summary drug response, how different tissue are responding to different compounds, and I can run this query to our database, and I see result immediately that, for example, pancreas tissue responding with these values, 8 and 0.2, to compound sorafenib. So these results can be used later for analysis, and we will show you how to use them in a minute. Also, we have this history here, so I don't need to put everything manually, and documentation about every node summary drug response, and you can search the documentation what fields to expect in your data. And also I want to show you that if users want to incorporate this query and this result into their software, they can use our open API. So here I show you 2 API endpoints. For this one, submission GraphQL is API for querying our Postgres database, and Guppy GraphQL is for querying our Elasticsearch database. So users can incorporate those API and queries to their code to run analysis programmatically. And other option for experienced users is to use our Gen3 SDK package, and we will show how to use it in a minute. So this package also helps to incorporate data discovery to their code. So to recap, there are multiple ways to discover data in Gen3 Commons: through exploration page with nice facets, through query page for more specific information, and open API and Gen3 SDK for programmatic access and incorporating data into your software. So with that, I want to introduce Fan Wang, our senior bioinformatician, and she will show how our researchers can conduct analysis in the Gen3.
Thanks, Victoria. Hello, everyone. I'm Fan from your Chicago team. I will lead you to analyze the cancer cell line data in a Gen3 data commons. And please feel free to ask questions at any time point during the demo. And today I will showcase the Gen3 service by exploring the cancer cell line encyclopedia, also known as CCLE data in the DCF sandbox commons. So here, and in this demo, I will show how the data be accessed, prepared, and summarized in a Python notebook, and then we'll briefly switch over to an R notebook for further visualization. And here you can see the landing page of the DCF sandbox. And if we click workspace, we can go to the workspace. And here we have the persistent drive where users can create files and they persistent after you terminate the workspace. So let me find the Python notebook demo. So here is a Python notebook. So first thing I will do is to install and import the Gen3 Python SDK. Next, we create the instance of the SDK classes. And first, we set the variable API to the URL of our data commons. Then we set the creds variable to point to the location of the credentials JSON downloaded from our user profile page. And if you're curious about where the profile page is, you can go to the top navigation bar, the profile page, and you can create an API key and download it from there. And then once you are back to the workspace, you can click upload and navigate to the file on your local hard drive and upload it to the workspace. Let's go back to the Python notebook. Finally, we create two Gen3 SDK instances, and the auth handles our authentication, and sub interacts with the data query, export, and submission. Now we'll use the Gen3 SDK to download the drug response summary data. And the SDK has a function export node that can be used to download all the structured data in a single node of a particular project. And here we ask for the CCLE drug response summary data, and we want to save it as a file and then read it in the Python as a pandas data frame. Now that the data is downloaded and we read it in pandas, and you can see that in this TSV file, it has a header for every property in the summary drug response node, and which includes the maximum activity, the compound name, AUC, which is area under the activity curve, IC50 and EC50, and also the submitter ID, which is a combination of cell line, tissue type, and also drug name. So the next thing I will do is to extract the tissue name and the cell line name from the submitter ID, and then add those as columns to our data frame. Next, I download a class of Python functions from a GitHub repo and load them into Jupyter Hub. So first, I download a file that includes some plotting functions. And then I run the file to read the class of functions into my Python environment. And finally, I create an instance of this Gen3 expansion class. So now my plotting functions are ready to use the defined properties from the data model that I like to plot. So here, the plotting part, we'll plot the distribution of the EC50 data for each tissue type using the Seaborn and the Matplotlib. And the custom function plot numeric by category, plot the distribution of the numerical property by each category. And here you can see I've defined the numeric category as EC50 and the category property as tissue. Then I run my custom function. And here you can see the legend part has many repeating colors because there are so many tissue types. And so the next function is to plot the only the top 10 tissues with the highest mean numeric values. And this indicates which tissue type is on average the most sensitive to each drug. And next, we plot AUC, which is the area under the activity curve for each compound and each tissue, but in separate plots. Yeah. And first, we still define the properties to plot as we did before, and then we run the function plot top 10 numeric property by category, which plots only the 10 category 2 values compounds. So each plot here is an individual tissue type, and then it plots different distributions for each compounds tested in that tissue type. And next, we create and save the summary tables of the data. And we just download it for use in the following R notebook demo. So first, we will create, we will calculate the number of tissue types and the cell lines tested for each compound. Then we calculate the number of cell lines for each tissue type. And finally, the data tables are saved to CSV files and read into R. Now that we saved our files, and we'll take a look at our summary tables. So the first one is the average drug response data by compound. So these are the average values across each cell line for each compound. And the second summary table is the average drug response data by tissue type instead of compound. And then finally, we'll create a table of the average drug response data by compound for each tissue. So finally, our data has been exported and massaged into a format for easy plotting, and our notebook can be created and use a ggplot library to make some interesting plots. And let's find the R notebook. So here is our R notebook. And you can see up here, first we read the drug response summary data exported in the Python notebook into R. And next we add a ggplot package to our library. And I also set some plotting options. Then we plot the number of cell lines and the compounds tested in each tissue type. So in this plot, we see that the number of cell lines per tissue type varies. However, roughly the same number of unique compounds were tested in each tissue type. So on the top is are the cell lines tested, and the bottom is the number of compounds tested. And the x-axis, we have the different tissue types. And finally, we read in the drug activity data to create this heat map of the average sensitivity of each tissue type and to each compound. So from this plot, we can see that each tissue type is most sensitive to the drug paclitaxel and panobinostat, so which are down here at the bottom. And they also have the darkest color, which indicates that their area under the activity curve is the highest, and meaning that the tissue are the most sensitive to these two compounds. And the X indicates missing data. So now we'll finish up the notebook demo, and let's hand it over to Dimitri. Thanks, everyone.
Okay, hi everyone. Can you hear me?
Yeah, we're good.
Yes.
Can you unshare screen? So can you see my screen now?
Yes.
Okay, good. So this will be just quick overview. How can you test hypotheses if you have some ideas? So what we have here, we have two commons, TopMed and Kids First. And so one of them is by a catalyst now. And we take adult data with atrial fibrillation, or AF. And from kids, we take congenital heart defects, CHD. And the idea was that it was shown that atrial fibrillation has some genetic background and nobody knows what it is. And people try to figure out what genes are involved in the condition. And for some people, atrial fibrillation can be developed like at teen age, 15, 16, you already can get it. And some people get it late in life, like 50s and 60s. And so the idea or hypothesis here was, for those people who develop atrial fibrillation really early, what's happening, they probably have some genes which are already affected in a way right from the birth, but you couldn't see the defect because it's not enough accumulated defects to see it. And this life, like you get like flu, some viral infection, or some toxins, which can add more genes which will be affected by those conditions. And then in some, all those predisposed genes and genes which mutation you acquire during the life can lead to atrial fibrillation. And so what we decided to do here, we just take gene panel from the Kid First and look only at the genes which are related to congenital heart defects. And the idea was that if you run whole genomic information that we have in the commons, it will take a lot of time and it will take a lot of resources. But once you are focused and you have like, at that point, we have about 300 genes. So you just take and test only those 300 genes. And so that's what we did. We take those from Kids First data, and then we apply it to another commons from by data catalyst, and to see what genes, those genes which are not supposed to be active in adult heart, whether we have any defect in them. And that's how we proceed this testing of this hypothesis. So we just annotate those genes and see what variants do they have using SNP-F and SNP-SIFT tools, which install in our workspace. That's what is nice about it. So you can combine both data in the same workspace. And then they just filter data only for mutations which are deleterious. So SNP-F will give you the information whenever you have a SNP, whether it's affect gene functions or don't. So we select only genes which were affected, and then we run a Fisher exact test and chi-square test. It's just statistic to see whether changes of that particular SNP in the patient has a different from SNP which changed in common population. And if there is no really difference, it's just physiological variant. So it has nothing to do with atrial fibrillation. And after all those filterings, we end up with 11 significant SNPs, which were common to both cohort, to Kids First and to TopMed. And then we'll just do PubMatrix search for those 11 SNPs and see what genes they are affected. And we found that one of those candidates is laminin genes, nuclear genes in the heart muscle, which is well-known atrial fibrillation related genes. So which means probably other 10 candidates might be considered as a good candidates. So here I'll just showcase how you can use two different commons and one common workspace to test your hypothesis. It doesn't matter how crazy your hypothesis sounds, you can just test it without much effort on the bench. So all data is there. So that's what I'm showing here.
Okay, thank you, Dimitri. I think we're going to hand it off to Alex, who's going to run through kind of the technical backend, because I know that about half of our attendees here are probably very interested in how we support the work that was just shown as an example of Gen3 Data Commons. And then I am, for the sake of time, going to forego much of my conversation at the end and just circle up on some of the questions that I've been getting in the chat once Alex is finished. So Alex, I'm handing it over to you.
Awesome. Thanks, Garrett. Hi, folks. My name is Alex Van Tol, one of the lead engineers at UChicago working on the Gen3 platform. I'm going to talk, go into a little bit more detail on kind of the technical backend that supports a lot of the features that you just saw from the other presenters. Specifically, I'm going to touch on authentication, authorization, data access, and then lastly, data exploration. So the first thing I want to start with is kind of the idea behind the Gen3 platform is to make sharing data easier. There's a lot of nuances in that, you know, we want to make sure that the right data is shared with only the right users. So we need to make sure that we're authorizing the users for the correct access. It's very common that data moves or is replicated across buckets or changes ownership, URLs change. We in the Biodata Catalyst project know that too well. So we have some stuff within the Gen3 platform that helps to make the fact that data moves a little bit less of an effect on data research reproducibility. So I'll touch on that. And then the other thing that is really important to Gen3 is making sure that we have an open API such that other platforms can interoperate with us. And so we have a lot of clients to our system. I'll talk about kind of the flow for a client application acting on behalf of users against Gen3's API as well. And so the order that I'm going to touch on things, I'm going to jump into authentication, authorization, hopefully get the drier but important stuff out of the way, and then I'll talk about data access and data exploration. And I'm going to touch on the Gen3 implementations for each of these. We have a microservice architecture, so I'm going to touch on each of the different microservices that kind of handle these different functions. And so the first one, and probably the most commonly known one for those that interoperate with our platform, is Fence. So this is our authentication and authorization service. For those that maybe are not familiar with this terminology, authentication is just who a user is. So this is usually like a username password combination, some way to prove that you are who you say you are. In the case of Fence, this is usually a Google identity or an NIH identity. And then authorization is just what you're allowed to do within the system. So this is usually what data sets you have access to. And so what we're looking at here is kind of a top level view of what's going on the internals of our system on the right-hand side. And then on the left-hand side, you can see a researcher interacting with the Gen3 platform through an API gateway. So most of our commons are deployed in AWS. So we have an elastic load balancer up front. We have a reverse proxy service running within our Kubernetes cluster. And that reverse proxy then proxies requests to the necessary microservices based on the actual endpoint that you're hitting. And so a typical user flow, as far as what goes on in the background, might look something like this. So first, that request is going to get routed to Fence. Fence is then so this is the page that you saw before. So if you're interacting with Gen3 using our data portal, this is the place that you would go to log in. So these buttons here are configurable per common. So in this case, Google is the only identity provider that's allowed in this commons. So from a user perspective, you would see this, you click login with Google. And then Fence is doing the OIDC, OpenID Connect handshake with the identity provider to actually log them in. And at that point, the user would be presented with a login screen. So depending on the identity provider, the login screen will look different. This is the user's browser is redirected to either Google or iTrust. Fence itself is fairly configurable, so we support a number of identity providers, including OrcID, Microsoft. In Commons, we support other fences as identity providers, and eventually we'll be adding the NIH Researcher Auth Service, or RAS, which is being talked about in a lot of contexts as an additional identity provider. And so after logging in, the user is back on that front page that we saw, where they can see some of the summary statistics and some links to different places within the Gen3 platform. One of the typical things that they would want to do at this point is maybe do some data exploration. And so we actually have a kind of a more complex policy engine that handles some more logic that Fence isn't fully capable of, called Arborist. So Arborist is populated with user information, user roles, their policies that they have in the system. And most of our services, including the exploration page, on the back end are using Arborist to either filter out results that users cannot see, or it's used to make decisions on whether or not a user should be able to take some action. So we're able to restrict certain parts of the system based on which user is actually logged in. And Arborist is the one that's kind of arbitrating those decisions on the back end. And just technical implementation details, Arborist is one of our only services that's written in Go. It's very fine-tuned and fine-grained in its purpose for making decisions based off policies. And so we went with something that hopefully would be a little bit faster than a typical Python implementation. And so not only does exploration, but other Gen3 services will also interact with Arborist for authorization decisions. Okay. The other thing that I want to touch on is just the fact that Fence itself is fairly configurable. So a lot of our commons will have specific identity providers that they want to support. We may get authorization only through the database for genotypes and phenotypes, dbGaP. So usually in a lot of these commons, the ERA Commons ID is kind of the user identity that's expected, because that's how we get authorization information. But Fence itself supports OIDC with any other identity provider that also supports that. And then in terms of authorization, so I mentioned dbGaP. So for most of these commons, when the data lives in dbGaP, we get the authorization information from dbGaP as well, in the form of these user access telemetry files, which list which ERA Commons IDs have access to which accession numbers or FIS IDs. So that's the main way in most of the commons, but Fence does also support a kind of generic way of syncing user authorization, and that's through a kind of specially formatted YAML file which lists user privileges. So we actually use this in tandem with the dbGaP sync in a lot of these commons for either developer access, developer level access to everything, or if the data doesn't exist in dbGaP, or it doesn't exist in dbGaP yet, this is another way that we can provide access to the data within the system that doesn't require having those whitelist files from dbGaP. So this is just a visualization of what I was just talking about in terms of the authorization sync. So usually we get stuff in the form of dbGaP telemetry files. We can also get this kind of user YAML. And then our user sync is a special cron job that we run in pretty much every hour in every commons to update the user access based on these two inputs. And then this is just showing that Fence and Arborist are both using Postgres as a backend. And during the user sync process, Fence updates Arborist with information about users and what policies they should have, so that those can be used for decisions later down the road. Okay, so going to jump into what it means for a third party to access data on a user's behalf, because it's pretty common that perhaps the Gen3 services that are being used may be limited to Fence, IndexD, and Arborist, which you may have heard us call our framework services. So these are kind of the foundational services that support data access. And so if we have a system like Biodata Catalyst, where we have a different platform that's being used for analysis, then that client application becomes an OIDC client of fence and is able to act on a user's behalf against the Gen3 APIs. And so we support this flow with just traditional OpenID Connect as per the spec. The main things that we have to establish are the user is who they say they are, the client is who they say they are. So there's some trust between Gen3 and the client, and that's in the form of a client ID and secret. And then the last piece is that the user consents to the client acting on their behalf within Gen3's system. Essentially, Gen3 sharing user information with that client application. And so long as all that happens and everything is approved by the user, then the client is able to go through a more traditional flow to get to the actual file objects. So this is genomic data, imaging data. Any of the larger unstructured files that we store is accessible through another service called IndexD, and I'll talk about that next. So IndexD is our data indexing service. Indexing in this context, I mean being able to locate data with easily used identifiers. So one of the common problems with data is that it moves, it changes ownership, it's replicated across different buckets. URLs where data is hosted change. And so what we're trying to do with the IndexD service, and when we say we're indexing the data, we're basically assigning persistent identifiers such that research remains reproducible even if the data itself moves around. So that's kind of the whole goal behind IndexD. The way that we do that is basically an abstraction layer over the object URLs where the files actually physically live in buckets. And that abstraction is in the form of this kind of JSON blob that contains minimal file metadata, including like MD5 sums, file sizes, file names, and then most importantly, the URLs that this file is located at. It may be located in multiple buckets. It's pretty common for things to exist in Google and also be replicated into Amazon, so that if someone's running an analysis in either of those, that they can choose the one that's going to cost them less and also be a little bit quicker in terms of access. And then the actual persistent identifier that we assign to these records is a globally unique identifier. So if you hear the word GUID thrown around, that's just the identifier that points to some logical piece of data. So the important thing here is that even if the data, if this bucket goes away and maybe we replicate into a different bucket, or the ownership of this changes and the data has to be moved to a different bucket, we can update that all within the record without changing the GUID. So the whole point here is to make sure that if a researcher runs some analysis using some GUID, that GUID should always point to that file object, no matter if it has moved or changed ownership. And then there's also versioning capabilities within IndexD, but for the sake of time, I won't jump into that. And so if we jump back into the data flow, so the actual communication that's happening between Fence and IndexD, so if we think of a typical signed URL request, given a GUID, Fence then communicates with IndexD to know the actual file object locations, and then Fence does the actual secure signing of that URL to return to the client application, such that the data can be streamed into whatever workspace or workflow needs to access it. Okay, I'm going to switch gears, talk about data exploration. So you may have heard mentioned, I think in Victoria's presentation, that there's a couple of different ways to query data in our system. So one of the important things to preface this with is the data that matches our data dictionary. So the data that gets submitted that matches our defined model for that system actually ends up in a Postgres database with kind of this mimicked graph-like structure. And we go through an ETL process to make that pieces of that data available in Elasticsearch, such that we can do kind of faster real-time filtering that you see in the exploration page. So Guppy is a service that we have that translates GraphQL and is able to do the search over Elasticsearch and return results. The other important thing that happens within Guppy, and Guppy is kind of what powers that exploration page that you saw, where you can kind of click on facets and dynamically filter results. One of the most important things that it does is the actual auth control. So it uses Arborist on the back end as kind of this middleware to filter out data that the user should not be able to see, that they're not authorized to see. And then the last thing that I'm going to show is this kind of architectural back end of what's going on. So kind of a lot visually. I'll direct you to kind of the bottom here. So as I was saying previously, the data that gets submitted into the system ends up in a graph-like structure in a Postgres database. And we actually run that through an ETL job to get it into Elasticsearch. And then our front end is using Guppy to actually query over this Elasticsearch service. However, we also expose APIs to search over the raw Postgres with GraphQL as well. So both of those are available. When we actually hand off PFBs, so in systems where we're generating a PFB and handing it off, we actually go back to our source of truth Postgres database and pull all the information from here to load it into that PFB. Because the Elasticsearch database doesn't have all of the data, it has the data that is available for filtering. And I am just about out of time. I will really quickly touch on interoperability. So one of the things that we're trying to work on is this kind of handoff mechanism with some standard serialization format. We have an implementation called PFB, which is basically Avro with some relational graph-like stuff on top of that. And so in the future, what we want to make sure we can do is generate a cohort, index it, make it available for future use and for referencing in whatever kind of publication is necessary for reproducibility, and then allow handing that off to different analysis systems. Okay, I tried to speed through. I think.
So I am gonna, if you don't mind going to the next slide.
Sure.
And then I promise this is the last slide that we're gonna show. What I wanted to do here is try to articulate this is not an architecture diagram. This is purely trying to think through how our different systems that Gen3 is a part of are communicating. And I want to use this slide just to address one of the questions that was brought up in the chat. There was a question around when we were showing workspaces and when we were showing the Explorer, how users interact. I think the perspective was coming from maybe the CRDC, but it's relevant to ask across systems how users are interacting with those services. And what I just want to call out is that's relevant to anyone who's working on interoperability is that Gen3 is implemented differently, but similarly in all of these 4 projects. So in all 4 projects, we have instances of what is commonly referred to as the Data Commons Framework services that Alex spent the foremost part of his presentation discussing, and that's things like authentication and authorization services and indexing services. Those exist in all 4 of these projects in very, very similar manners, which is great in driving some of the interoperability contexts that we're working through. What exists notably differently between the 4 projects is how we use that front-end component, so that Data Explorer, those workspaces. Those are available in projects like Biodata Catalyst and in the future in Anvil for data search, et cetera. But are not used in the Kids First and the CRDC projects where we're relying on data portals that are in some cases homegrown for the specific project and in other cases pulled from previously existing systems. So that's the kind of the difference that I think is just relevant to keep in mind as we're having conversations around interoperability and as we have a lot of these PFB handoff mechanism conversations, that how we address those as the University of Chicago can differ depending on the project. And so while we'll always strive for standardization, there is kind of a need when we're talking across projects to consider how things have been implemented. I'm going to stop talking now rather than going to my next slide because Brian spoke to a lot of what I wanted to speak to. Mo, I'll take your lead. We can either take some questions now, or if we're way too over time, I'm happy to join one of those rooms I think you just sent a list about.
Let's skip questions now. We're a little behind time. As Garrett mentioned, there is a virtual Q&A and a spreadsheet for signing up for the time slots on the agenda. I'd like to hand it off to Alex Bauman for the Terra part.